Hey guys, thank you for coming. Yeah, my name is Shannon. I work at the Chan Zuckerberg Initiative, and today I'm going to talk a little bit about a project that I've been working on for about the past year called Starfish. I'm going to talk a little bit about the science behind the project and the reasoning for why we made it, and then I'll go into a little bit more technical detail on some of the aspects of the library. So let's get started. So first things first, if you're like myself and haven't taken biology since high school, we're going to do a mini biology refresher on some of the key concepts in this presentation. If you do know a lot about biology, I apologize. But so the central dogma describes the flow of genetic information from DNA to proteins created within your cells. And it basically states that your DNA makes RNA, which makes proteins. And when a particular portion of your DNA creates a particular type of protein, that's called gene expression. That gene is being expressed. So the set of DNA that you have in your cells is the same in every cell, but the types of proteins that are getting created in your cells, meaning the types of genes that are being expressed, differ across cells. So you might have one set of proteins being created in your skin cells and a completely different set of proteins, meaning genes being expressed in your brain cells. So biologists were able to map human DNA in a project called the Human Genome Project. You guys have probably heard of it, ended around the early 2000s. But they want to take research a step further. They want to understand why, if all the cells in your body have the same DNA, do they behave differently? And they think the key to understanding those differences is the difference in gene expression between cells. So there's this effort going on to map every cell in the human body based off of its unique gene expression. And this is the effort that the Starfish project is aiming to support. So how are biologists doing this? How are they profiling cells by gene expression? There's kind of two general approaches right now. One is this one on the top here, this RNA sequencing approach. So what happens here is you start with a tissue sample from somewhere in the human body. You remove cells from that tissue sample and kind of expand them to extract RNA molecules that you then run through the same type of sequencing technology that was used in the Human Genome Project to give you this end result, which is normally some type of cell by gene table that describes the cells that were in your tissue sample and the genes that were being expressed within each cell. So this approach works super well. It's really popular. But the one downside is that by removing the cells from the tissue sample, you lose the spatial information about where each cell was relative to each other. And biologists believe that this spatial information matters. So to give you one example, there's a ton of research being done into cancer cells, into why certain types of cancer cells resist treatment when others die off. And there's a hypothesis that the difference has to do with local interactions between different cell types. But there's no way to confirm this hypothesis if you're able to profile cell types but then not know which cells were next to each other. So to solve this spatial problem, biologists created this new technology called image-based transcriptomics. So with this approach, you have your same tissue sample, but instead of removing cells from your tissue sample, you instead inject fluorescent probes into the cells that bind to RNA molecules, and then you take a bunch of images of your tissue sample and run those images through an image processing pipeline that gives you the same cell by gene information you would get from your RNA sequencing approach. But in this approach, since you're just analyzing static images, you're also left with the information about where each cell is within the larger context of your tissue and what types were next to each other, if there's clusters, good information to analyze. So this is an emerging technology, and it's what we're going to continue focusing on. So what does one of these experiments look like? The general approach is they start out with their tissue sample. They isolate a specific subsection of it because they can't observe the entire thing under a microscope at once. So they isolate a subsection of it into a field of view of the microscope. And then they attach their multicolor fluorescent probes. So these probes are created to bind to specific RNA molecules. So the idea is they bind to these RNA molecules and then they emit bright spots. So if you're observing your tissue sample and you maybe see a bright red spot, that indicates the presence of a specific RNA molecule. So once your probes have bound, you then take images of your field of view in every corresponding color channel to capture the spots. So biologists design these experiments so that they know that the presence of a spot in the same location across a sequence of color channels maps to a particular type of gene that they're targeting for. They design the experiments to be this way. So a spot in the same location in, say, the blue channel and the green channel will map to a specific gene, whereas the red, yellow, and green maps to a different one. And this is just a really small example of what this process looks like. In reality, biologists have to do many rounds of this color probing and imaging to capture the number of genes that they're targeting for. So this gets repeated a number of times on the same field of view. And then everything gets repeated on every field of view in your tissue sample. So what you end up with is a ton of images, anywhere on the order of 100 gigabytes to 10 terabytes of images that just represent a simple tissue sample. And then you need to run those images through an image processing pipeline to give you your final cell by gene table. So what does this pipeline look like? The problem is pretty straightforward. At the end of one of these experiments, you have a stack of images with a bunch of spots in them, and the problem is basically find all the spots and then associate those spots to genes that you were targeting for in your experiment. So maybe you'll first need to register your images to align them in physical space, run some filtering to reduce background noise, make your spots brighter so that when you run a spot finding algorithm, it works well. And then you do this decoding gene identity step. So that is the bit that I talked about the biologist knowing the sequence of spots across color channels that correspond to a gene. This is where they provide that information and where we assign a particular gene to a particular spot. So once you have that spot to gene mapping, the last thing that you need to do is then just assign those spots to cells within your image. So this bit is a little bit more involved. It involves cell segmentation, so that's outlining the boundary of the cells in your image, and then just assigning spots within the boundaries of those cells. Once you've done all that, though, you're left with your final cell by gene table, which is the goal of one of these experiments. So the problem is straightforward. It's a straightforward process, but the problem is that everyone's doing it a little bit differently. So on the left here, I have a few examples of experiment types that my team has worked with. So not only is the biology slightly different in these experiments, so the color probes or the types of probes that they're using might be different, or the order in which they're probing and then imaging is different, but the way in which that they want to process their final images is also different. So even though two groups may want to do a filtering step as the second step of their processing pipeline, they want to use two different filtering algorithms to do it. Same goes for many other steps in the pipeline. You can see here that each group takes a slightly different route through this processing pipeline to the end goal. And then on top of all of that, they're using a variety of different tools to create these processing pipelines. So on the bottom right here, I have a few examples of the common tools that labs are using when they're creating these image processing pipelines. And this creates a super segmented landscape, and it makes it really hard to compare not only the results from these pipelines, but the pipelines themselves. You know, like this is an emerging technology, and people are really interested in understanding what types of algorithms work well on this type of image data, and it's very hard to do when it's all written. Everyone's pipelines are written in a different language, and they use different file formats. It makes it harder to advance the field. So to solve this problem, we created the Starfish project. We thought that this technology was really awesome, and we wanted to accelerate the adoption of it, and we thought that the best way to do that was by trying to standardize the field a little bit more. So Python is an open source Python project in which people can build their own processing pipelines for their imaging data. It provides a number of different algorithms that we just saw, a variety of different algorithms that we just saw on the last slide, and a variety of different steps that someone might need in their processing pipeline. And by creating this, we're hopefully doing it in a really usable way and making it scalable. We have the ability to standardize the inputs and outputs of this library, and we're also creating a community in which people can collaborate and share their ideas about these pipelines, share their results, share their algorithms even if they contribute to the library. So that was the reasoning for the project, and this is the team. All right, so I'm going to talk a little bit more about the library now, a few aspects of of the Starfish library. So I'll talk a little bit about our object models, our file formats, and then our processing model. So object models in Starfish. The main object model that we're using is what we call an image stack. It represents that field of view, that stack of images for a field of view. So it's a 5D tensor that's backed by the library Xarray. And if anyone is unfamiliar with Xarray, it's very similar to NumPy, but you can label your axes and add a bit more metadata information, which is important for us when we want to maintain things like physical coordinates throughout our processing pipeline. So we use it pretty widely. So you have your image stack. It's your main object model that someone's going to use for processing their field of view for registration, for filtering. It's what they send into their spot finding algorithm. But then the output of a spot finding algorithm is what we call an intensity table. It's also an Xarray-like object, but it's a 3D tensor that represents spots found across rounds and channels of your experiment. So once you've found your spots, the last thing, or the second to last thing, I guess, you need to do is assign those spots to genes, and that's where your codebook comes in. So a codebook is an object representation of that series of spots you need to see across your image stack to represent a specific gene. So on the bottom here, you can see that one entry in your codebook represents the series of spots that I would need to see in the same location in my image stack to represent this MATLAT1 gene. So you provide a codebook and an intensity table to your decoding algorithm to get your decoded spots. And then the last thing you just need to do is assign those spots to your segmented cells to get your final cell by gene matrix. So a few of the file formats that we're using for Starfish. We try to make the file formats as simple as possible, as readable as possible. We use JSON a lot. So to represent these image stacks, these fields of view, we use NumPy or TIFF files. It's a collection of NumPy or TIFF files that's organized by a JSON file. Codebook is just a JSON file, and an experiment, which describes where all of your images are for your entire experiment, your entire tissue sample, is just a JSON file that describes the location of every field of view in your experiment. The outputs of Starfish are a little bit more flexible. The main output is a netcdf file for an intensity table, but the expression matrix, which is that cell by gene table, can be outputted in a variety of different formats depending on the use case that someone's trying to use the data for. All right. Processing model. So if someone's trying to use Starfish, someone's coming and they're trying to analyze their data, the first thing that they're probably going to want to do is work on a small subset of their images to try to tinker around, play with different algorithms and steps to see what works best for decoding their spots and what works best on their images. So the idea is that someone's going to work with maybe a single field of view to determine what works best for them. One step in a pipeline in Starfish is what we call a pipeline component. Within a pipeline component, you have a variety of different algorithms to choose from. So you can see here that on the filtering pipeline component, this person has chosen the Gaussian filter, and on the spot finding pipeline component, they've chosen a local search algorithm. So someone's going to play around with everything Starfish has to offer, figure out what works best for them. What works best for them is what we call a pipeline recipe. So a pipeline recipe describes the sequence of steps that they want to use in their pipeline, along with the algorithms within each step that they've chosen. And I want to highlight here that we're not writing a lot of these image processing algorithms ourselves. We try to leverage as much from the open source community as we can. So a lot of our image processing algorithms are scikit-image algorithms that we've pulled into our library and added multi-processing and logging into them into the library, wrapped into the library. Yes, so I want to highlight that. All right, so once somebody's figured out their pipeline recipe, they've figured out what works best on their data, they then want to apply it to their entire experiment. So this is where a pipeline runner comes into play. A pipeline runner is a workflow executor that runs in the cloud. The idea is that you send your experiment images along with your pipeline recipe into the pipeline runner, and the pipeline runner distributes the processing of those fields of view in parallel in the cloud. And then once each field of view is processed, you concatenate all of them into one giant cell by gene table for your entire experiment. So what's really nice here is that by parallelizing this processing, we've sped up the processing of this data a lot. So something that has previously taken labs hours, if not days, to process can now take 20 minutes in the cloud. The pipeline runner is a language agnostic. It can be written in a variety of different workflow languages, and that's the bottom here, just a few, and the ... is many more. Yeah, so that is processing at scale. So I have just a small code example here, which hopefully you guys can read. This is a pipeline recipe for an experiment called Marefish. You can see here that they do a number of different filtering steps, and then they do a pixel spot decoding algorithm. And so what's really nice about Starfish is if the Marefish folks decided, oh, they wanted to play around and try, different filtering algorithms, it would be as easy as just, swapping a few lines of code instead of having to export their data and open up a whole new tool, learn a whole new language, something like that. So this is what we're really trying to provide to the community, is this ease of experimenting and hopefully a readable API and, yeah, making it really easy for them to explore. So a couple things that the team is excited about exploring in the future. We want to look into GPU processing to see if it speeds up some of our processing steps immensely. We've explored using some multidimensional chunked file formats like ZAR to represent the giant images that are coming in. And then machine learning algorithms. So there's a few areas of the pipeline where there's some really cool machine learning research being done. For example, that cell segmentation problem, that outlining cells in your images problem. There's some really cool research being done on applying neural nets to solve that problem better. And it would be great if we could incorporate some of those algorithms into Starfish for everybody to benefit from and to see if that drastically improves the segmentation portion of the pipeline. But that is it for me. Thank you so much for listening to my presentation. Yeah, answer any questions, or you can email the team, and that's our GitHub link.
Questions?
Thanks for the talk. You just kind of mentioned this very briefly at the end, that you're interested in the cell segmentation and identification. So does Starfish actually do cell counting for you as well as measuring the activity, or are you just basically trying to identify cells for some of the downstream steps?
You mean counting the number of cells in your images?
Right, yeah.
Yeah, I mean, you would get that inherently from the final table that has, all the cells that were found and all the genes within the cell. So yes.
Okay.
Yeah.
And sorry, the platform, this Transcriptomics platform,
yeah.
Are you familiar with the Codex platform?
I've heard of it, not super familiar. But...
Okay, so in that case,
okay.
Other questions? Getting my exercise today.
So speaking as someone who's written a lot of pipelines and often kind of ad hoc pipelines, I'm just wondering if you've tried to standardize on a standardized pipeline like Kubeflow, which is where I see myself going with my own work, wanting to take my ad hoc work and map it into something that's, I don't know, maybe a little more future-proof. I wonder, have you looked into Kubeflow or some other, the great thing about standards is there are so many to choose from. Have you looked into standards for pipelines?
I'm not sure I understand your question. Are you talking about like a standard pipeline for this type of data, or a standard processing pipeline?
A standard set of tools.
Standard set of tools. I guess we would hope that Starfish would be the standard set of tools for this community. I mean, yeah, we've looked into. We try to use as much as possible from existing technology, but this image data is pretty specific, and so the standard needs to be created, I guess, is the problem that we're trying to solve. Yeah. Yeah.
Hi, I really like cartoons you made to make the concept really stand out. Do you happen to have like maybe a live demo you could do for just?
Oh gosh.
Small thing? I
Live demo. I don't have a live demo ready. I could show you some of the code, but no, I'm sorry, I don't have a live demo ready.
Maybe figure that out offline.
Yeah, I'm happy to show you offline for sure.
Anyone else? Other questions? Got plenty of time. Okay, let's thank our speaker again.